Hypophysation, or other known as induced breeding, the artificial process by means of which the extract of the pituitary is introduced inside the body of both due-matured male and female fishes. Then, after being excited, they lay eggs in the pond water and subsequently fertilization takes place, and the process is called induced breeding of fishes. This kind of breeding is otherwise known as hypophysation. Additionally, induced breeding is a technique where ripe fishes are stimulated by pituitary hormone introduction to breed in captivity. This promotes timely release of sperms and eggs from ripe gonads. In simple words, spawning in fishes induced by some artificial breeding stimuli may be called induced breeding. The active factors like LH and FSH hormones are responsible for induced breeding. History of induced breeding. The techniques of induced breeding was first evolved in Argentina after producing pituitary extract by scientist Biehase in 1930. Brazil was the first country to develop a technique for hypophysation in 1934. By von Ihering and his team successfully breed a catfish with pituitary hormones, and hence credit for the present-day concept of induced breeding of fish goes to Brazilians. Russians applied this technique to breed sturgeons. In India, the first attempt to induced breeding was made by scientist Hamid Khan in 1937 on Serhinus mrigula. Chaudhary applied this technique in minor carps like Esmus danricus in 1955. Ramaswamy and Sundaraj, 1955 to 1956. First induced to breed Clarias batrachus and Heteronusts fossilis. Chaudhary and Alkuni, 1957, succeeded in inducing L. rotata, L. bata, C. mrigla, C. Reba and P. Sarana Parmeshwaran and Alkuni, 1962, successfully breed to exotic Chinese carps like grass and silver carps. Chonda, 1984-1990, described a method for the mass scale and breeding of IMCs and silver carp. Advantages of induced breeding. There is no possibility of mixture, and as a result the pure form of fish seeds are obtained. Large numbers of eggs are available from a fish through induced breeding. In the same season, like carps for example, can breed more than once. Can produce hybrid fishes. Economical. Structure and location of pituitary gland. Pituitary lies in a well-protected depression myodome on the floor of a cull on the ventral side of the brain near optic chiasma just below W, the hypothalamus. It contains gonad-stimulating hormones LH and FSH. It is more or less round in carps, oval in cattle and roe and bear-shaped in mrigal. The pituitary is located in a concave cavity known as sella turcica and enclosed by a thin membrane known as gyromata. It may be attached to the brain by a short stalk called the infundibular stalk. Types of pituitary glands. Based on the presence or absence of the stalk, the pituitary is classified into platybasic pituitary or without stalk. For example, murrels and glassfish, Ambercis species. Leptobasic pituitary or with stalk, just like carps and catfishes. Procedure of hypophysation. It is collected from the mature fishes. It is located on the ventral side of the brain. The head is dissected out, expose the brain, and then isolated from the ventral side and stored in alcohol. preparation of pituitary gland. The gland are mixed in a tissue homogenized with a little distilled water. The homogenate is diluted with distilled water. The preparation is centrifuges at about 1000 revolutions per minute for 5 minutes. It is preserved in glycol. Selection of breeders. Mature males and females are selected for hypophysation. 2 males and 1 female form 1 unit for hypophysation. The extract is injected intramuscular at the base of the pectoral fin or causal fin. The selected males and females are kept on a table on a cotton cushion. About 0.5 to 2 milliliters of extract is given for breeders weighing up to 10 kilograms. For female, they are injected 2 doses, namely, a preparatory dose and a resolving dose. Duration between these 2 doses is 4 hours. For male, 1 dose is given. breeding. After injection of the pituitary extract, the breeders are introduced into Hypo. The breeding Hypo is rectangular mosquito net cloth enclosure. It is suspended in a pond water with help of casuarina poles. Running water is preferable. The mating takes place. They breed in 6 hours. Hatching. In 10 hours, the eggs are fertilized. The fertilized eggs are transferred to a hatching Hypo. It is made of up to 2 rectangular mosquito nests. It is suspended in water. It consists of an inner Hypo and an outer Hypo. The hatchlings are kept in the outer Hypo and kept for 3 days. Finally, they are transferred into nursery ponds.